Yeah, I see another thumbs up. And yes, thank you so much. Okay, let's get started. So I'll be presenting on behalf of our lab about one of the projects and software at this point, quite a mature software we are developing in our lab. It's called segmentation for microscopy or microsam, wherein the idea is simple. We want users to be interactively able to segment stuff. What does all that mean? So let's say our idea is directly inspired from Segment Anything. This is a model which was devised or let's say led by the development from Meta AI, wherein the idea for them was to develop a model for interactive segmentation. And as you see over here on the left, a user is, let's say, having the possibility of interactively segmenting objects, wherein you put a click or the hover blue thing you see is basically an input prompt, which is a point. And then you drag it on the object of interest, try to segment it. And let's say if you precisely want to delineate an object, what you do is put a negative interaction saying precisely, this is the frog I want to segment. And the snail is something which I do not consider. On the right side, very quickly, it's the automatic segmentation method, wherein the idea is instead of putting one grid with any manual intervention, you just put a grid of points on the entire image, segment all the corresponding objects, and do some top-down post-processing, mostly post-hoc, to get the best objects out of it. Now, why is the hype over there? Why do we care about it essentially? So, the first thing was the interactive segmentation, wherein the message on a higher level was that you, as a, let's say, interactive segmentation user, could segment almost any and every object, regardless of their morphology or let's say where they lie inside the image without any annotations at all. So you put in a point, you drag a box around your object of interest, and you can get the segmentation out of it instead of literally painting through the entire cell, for example, which is an interesting question for us. So what they use is something what is called prompts, which is a point or a box, or sometimes they have an option of putting masks, which is at least in an annotation point of view, not necessary or interesting for us either. And in general, as a rule of thumb, if the objects are more fine-grained, the more prompts you give as, let's say, a precise idea of what an object could look like, the better the predictions get. The model is quite versatile because even if you go, let's say, zoom out and go beyond the interactive segmentation pipeline, you can use, let's say, this model as an object detector in a semi-supervised manner, wherein if you have an object detector which can generate bounding boxes, for example, you can get the corresponding segmentations without training like a pixel precise classifier, for example. You can also derive nucleus seeds or have, let's say, something what is called spots, and then try to derive, let's say, segmentations out of it because those are considered as points, or let's say in 2D space, it's a Y and X point, for example. The model is completely open source. And how did they really train this entire model? So without going into too much details, there is 2 points for their success. The first pointer is they had a lot of images, or they collected a lot of images, iteratively train this model to, let's say, simulate how a user would segment objects and try to, let's say, improve this heuristic one by one to, let's say, train on, let's say, 10 images and then scaling it up to 150, 500, something like that. And that's how they generated something around 11 million images, which are like densely annotated. Um, Now, what made us curious is how does it work for microscopy? So on the left side, you see interactive segmentation or cell segmentation in phase contrast microscopy. What we observe over here is for cells which are isolated, it does a very good job. However, cells which are in a cluster somewhere, there are some troubles of identifying cell out of the cluster, for example. And on the right side is the automatic segmentation, which we briefly discussed before, the grid of points and doing a post-hoc processing. Now, the bigger question we started to ask is, this is working pretty good, I would say, but can we really improve this further for microscopy? The answer is yes. So on the left side, you see, regardless of how, let's say, sparsely or densely clustered the cells are, the segmentation using a point over here does a very good job. And on the right side, there is a bit of a architectural change we had to make the automatic segmentation a bit more meaningful. But this is how a complete instant segmentation for identifying cells would look like for us. Now, what is segmentation for microscopy and what did we do over here? So we extended the model architecture, as I mentioned, for better automatic segmentation. We re-implemented the entire SAM training procedure and extended it so that a user has the flexibility to, let's say, train one model for interactive and automatic segmentation. And we extend the functionality of segment anything to volumetric segmentation and tracking. This is an important mention because implicitly, segment anything is a 2D model. And what we do is basically do some silly, let's say, segmentation and stitching on top to get the 3D segmentation and tracking outcomes or tracklets precisely from the same trained model we have. And finally, we trained two big models. One model was for light microscopy, device for cell segmentation and nucleus segmentation, and another model for electron microscopy for, let's say, segmenting organelles, which kind of look like spherical or roundish or potatoish in structure. The reason we could do this or went for this is because that's the only heap of data we could find. So that was the motivation to, let's say, get a good organelle working model. And it does a pretty good job for mitochondria and nucleus segmentation. Now, what are the improvements we observed? So let's quickly hop through this. This is results with interactive segmentation on different tasks, different data sets. And we observed that once the model is trained on, let's say, a set of images, it of course does a good job on the left side, all these four images. But on the right side on images, it has never seen, for example, H&E stain histopathology images. It still tries to recognize the, let's say, morphology of the object of interest at hand, which was pretty interesting for us. And this kind of gave us out a hint that these foundation models could really pave a path for microscopy image analysis in our context, especially. Another thing was automatic segmentation, which I previously briefly explained. So the idea is we stacked an additional decoder head on top, the ones who are interested in the technical details, to make automatic segmentation possible. Now, without going to details, there are EM and LM results shown over here. But the idea is to literally identify all objects in the image, which Microsam, from our experience, did a pretty good job in these cases. And what is our overall contribution and why are we here actually? So we are here to talk about and play with Microsam, which is our Napari plugin, which enables you to do a few things. The first thing is automatic and interactive segmentation in 2D, the same in 3D, and automatic and interactive tracking in 3D, in 2D over time precisely. And let's say all these features, what we have provided you does not work for your data. What you can do is train your own model, preferably using the Python scripts. We talk about these tiny details in a second. And once you have your data, some annotations generated, let's say with Microsam, Cell Profiler, Fiji, QPath, any of the annotation softwares, then you can train your own model on, let's say, an instant segmentation or a semantic segmentation task. And the core functionality for this is we provide support for the original segment anything models and our foundation models, how we call it, or generalist models. The other thing is enabling multi-dimensional segmentation, which is of a huge interest in our lab as well, and allowing tile-based prediction for larger images. Because if we treat a large image as a full image and feed it to the model, let's say the resolution of the objects would change the, let's say, subjective target of interest, which is not what we want to do. If you want to segment cells, we want to segment cells individually and then, let's say, do it over tiles or a field of view where cells are observable and then stitch them back together. Um, Now, a quick teaser, like we've been talking about this in a few conferences here and there, but a quick teaser for everyone. We are working on Microsam 2 right now. And this is kind of directly inspired by the new version of Segment Anything model, which has been out for a year now, actually. Wow. And the model can universally do image and video segmentation. So with the click and box logic, as we discussed, they can do exactly the same for videos as well now. So that is pretty cool. And they showed some preliminary examples on the right side for microscopy images as well, which, let's say, led us to think, can we improve this for multidimensional data? Or let's say, to simply wrap it up, can we create a universal microscopy segmentation heuristic? Let's say, according to our preliminary results, the answer is yes. And the idea is right in front of you, regardless of the input, which is 2D or 3D images, it will be accepted by the Microsam 2 framework. And as a user, you could do interactive segmentation or automatic segmentation with the same model. So you do not have to do the, let's say, fancy post processing on top to stitch things back with some heuristics, but rather do everything end to end with the spatial context in 3D, which is kind of very critical. A few preliminary results over here, which I presented in another conference. On the left side, you see is interactive segmentation, wherein we're trying to segment nucleus and mouse brain, if I'm not wrong. And on the right side, it's data from the Lycon cohort. If you guys are familiar, it's doing connectomics with light microscopy in a nutshell. And they want to understand how these neuronal cells connect in these images. And what we saw is, with the strengths of this model, automatic segmentation is something which works pretty well as well on this data. And the fun fact for both of the data sets is, this is one universal model which works on light microscopy and electron microscopy. So we want to really go towards the universal aspect a bit more, which is why we trained one model and tested it on two data sets which it has never seen, for example. Now, all being said, what is, let's say, the workshop overview right now? So I'll give you a few examples and you can choose what to, let's say, work on. And let's say, since I didn't communicate this well before, what I mean by work here is, you can ask me questions, we can do things together, we can brainstorm, chit chat, do stuff. But the idea would be, I show you one example for 2D segmentation, one example for 3D segmentation, but still with Microsam 1. And we see a few things around fine tuning, how the documentation looks like, or if you have any questions, if you have any problems which you want to chat about, we would be happy to take it further from there on. Sounds good? Any questions? Because this is the last slide and would be a good time to take in some questions. So I think you guys can unmute yourself or drop in a question in the chat. But then I would probably stop sharing and then give you guys a few minutes for questions. Meanwhile, I get set up.
Hi. So I have been setting up to.
Yeah, hi, Philip.
Hi, how are you doing?
I was like, how do I know you?
Yeah.
I'm good. How are you doing?
Yeah, very good. How are you? I've
Cool,
been
pretty
using
good.
Microsam a lot, and at the
Yeah.
moment I am building a fine-tuned version for our lab. So the idea is, we take all the cells we typically work with and the imaging modalities we typically work with, and
Yeah.
I segment a lot of these images, and then we
Yeah.
make one that's perfect for like our lab. So a few questions. So the first one is, my instinct is I can mix different modalities as
Mm-hmm.
long as I take at least like a large enough vision transformer, like the one of the L models, that it's robust enough to take that. What do you think?
Mm-hmm. So is there a reason why you're opting for the size more than the data aspect? Or it's just an intuition you're going with?
So it's firstly, it would be practical for everyone in the lab because we might
Mm-hmm.
use different modalities.
Yeah,
But
yeah, yeah, yeah.
secondly, I'm also a bit interested from the research perspective. How
Okay,
well will it deal with different modalities, or can it generalize to
okay,
unseen modalities and so on?
okay. Mm-hmm. I mean, my answer here would be the first one would be on the vision transformer part.
Yeah,
There are four sizes of vision
yeah,
transformers completely, or three, let's say, to
yeah,
simply put it. From segment anything, there's base,
mhm,
large, and huge.
yeah,
And this is the order, base being the smallest and huge being pretty
yeah,
massive. And the idea is the bigger the model is, let's say the more time it takes on your device to do segmentation,
yeah,
because it's literally processing every chunk throughout the funnel,
yeah,
throughout
yeah.
the model as a funnel. So the first answer would be, I would say, if there's no specific reason, I mean, if you want to explore a research question, go
Yeah,
for it and try all three models.
yeah.
Otherwise, if you are more focused on solving a problem, I would stick to the base model
Okay.
because it does a good job. It's
Related
quite
to that,
efficient. Yeah.
um, have you experimented with doing distillation? That maybe I train a huge model and then I distill it to base size model?
So usually this is something which is possible. We
Yeah.
haven't tried it yet from our
Okay.
side because our research questions were always focused on solving a biological problem where accuracy was in demand.
Mhm.
We didn't care about other things. I mean, there are questions we care about in parallel,
Yeah.
but sometimes with biological questions, we really cared about the accuracy. So
Mhm.
in those cases, at least off the top of my head, what I remember, trying out some other things here and there were not as good as like literally retraining the model on the
Okay.
data.
Yeah.
Yeah. And coming back to the data question, very quick answer would be just go for it. Go for as much data as possible.
Mhm.
Segment anything, or Microsam likes it actually. So if you have a lot of diverse data, that is kind of a key goal. Because if
Mhm.
you have, I don't know, live cell images, because I remember you have phase contrast live cell
Yeah.
images, right? Yeah.
Yeah.
If you can pull on more data from around and just throw into Microsam, Microsam would definitely generalize well for not just your data, but maybe new incoming data in your lab as well.
Yeah. Cool. Cheers.
Perfect. You got it. There's another question from Vasant. Did you all do any segmentation on bright field images? Yes, we tried this as well, actually. And let's say for most cell segmentation and nucleus segmentation tasks where let's say the membranes were kind of obvious, the model did a pretty good job. But for some other cases where some cells were overlapping and strange things were happening, yeah, that's where some discussions begin. But I would say on higher level, it does a good job for segmentation and bright field as well.
Okay, yeah, thanks. No, we've been trying out a few on our own, but not had much luck with it. And I think more work needs to be done. But just curious if other
Okay.
people are trying
I
the same
mean,
thing.
if you have any question, I try to be very active on ImageSC. So if you have
Okay.
any questions, just shoot me like an email, ImageSC, an issue on GitHub, and we look at your images. That
Yeah.
should not be an issue. Yeah.
Yeah. Sounds good. Thank you.
Perfect.
Yeah.
Okay. Any more questions, guys? I mean, you can still think about them, but let me then prepare some stuff meanwhile. Okay. Yes. Okay, if there are no questions, I would probably get started with the next part by giving you a quick intro about a few things. Is my GitHub screen over here visible? Yeah,
Yeah.
perfect. Thank you so much. Okay, cool. So this might seem like cheating, but it's not because this is from last year's I2K, wherein we went to Milan to give this in person and gave this online as well. So this is kind of a similar setup of the workshop. So if you have heard it, I'm sorry, you can go ahead with more questions. I am open to that. But for everyone else, I'll give a quick run through on how to work with things. So before getting started, Microsam, let's say as a package, is deployed in CondaForge. And you can install this quite easily using Conda, Micromamba, Mamba, whichever, let's say, package manager you prefer in the CondaForge ecosystem. That would be an important tip. And there are, let's say, a few guidelines. I can also share you these links later on or add this to the workshop table for segment anything for microscopy. And then there are a few tips and tricks over here on what you can do with the example data or open source example data we have provided. So let's go to the console first. So let's open a new terminal and see how things would look like. So let's say if you were doing this yourself, what you have to do first is go to Microsam, then workshops and I2K 2024. Then you will see a bunch of scripts over here. There's a README file over here as well, which kind of details down on what to do as each and every step to, let's say, get you started. But you have to trust me here. We do some things together. I will activate a pre-installed environment using Micromamba and then open the Napari console to, let's say, get your feeling of how the GUI looks like. Okay. So the first thing is this is how the Napari GUI as an interface looks like when you just open Napari bare bones. So the first thing what we do is go to File, go to Open Sample, and you will see something as a console here, which is called Segment Anything for Microscopy. We have provided a few example data sets for you guys to get a feeling of what you're trying to segment and then hop on your data in case you are in doubt, for example. We hop on to, let's say, one of the 2D images. Let's go for this because you might have seen a few examples from this, so it would be much more comfortable to explain things on this one. Okay, so this is one of the images. Now we open the Microsam plugin. So if all the installation things went well, you will see a widget over here called Segment Anything for Microscopy, wherein there are a few options over here. To quickly walk you through, the first one is annotated 2D, which might not be so obvious, but the idea is you can do annotation only on 2D data with this annotator. The next option is 3D, the same reasoning, but for 3D data. The third one is annotated tracking, which helps you track over multidimensional images or videos precisely. Then there's image series annotator, wherein you can annotate multiple images in one stroke. There's a new, let's say, work in progress effort, which is called object classifier, wherein the idea is once you have developed or got your instant segmentation outputs, can we, let's say, discriminate if these cells look similar than the other pool of cells, assuming that there are two cluster of cells in my image. I won't go into too much details because we are preparing the manuscript right now for this. But if you want to check it out, we have documented this in our official documentation. If you have any questions, just hit us up. There's a fine-tuning widget, which is honestly not so intuitive. So I would always recommend going for Python scripts, wherein you have a bit better feeling of what you're doing. Otherwise, if you're not so comfortable with Python scripts, we have our command line integration scripts, wherein just with the command line, you provide your images and it trains a model for you as an output. And there are some miscellaneous settings. Let's not go into the details. So since the image over here in front of us is a 2D image, I click on annotated 2D. On the right side, this will open a segmentation widget for us. Let's wait for a few seconds. Whenever you see Napari is not responding, just wait for it. It's a bit, let's say, wonky on some sides when we are trying to load some heavy dependencies, but you just have to be patient with it and just let it do its thing. And there we go. Nothing crashed. Everything worked out fine so far. Now we, let's say, run the default settings. So over here is the image layer, which selects the particular image. There's a collection of models over here to very quickly go through them. This is the original model for natural images by Meta AI, which is the segment anything model. And we, from our side, have a model for light microscopy, electron microscopy, and two follow-up works for medical imaging and histopathology as well, which really go in the direction of trying to create a foundation model for medical imaging or bioimaging in general. There is a big console of embedding settings. We won't go into details on that. There is an important parameter for people who would be doing large scale image analysis, which is called tile shape and halo. And this is basically doing the tiling based prediction over the entire image. I see a message, I will keep on answering them. Is this applicable to fluorescence images? Yes, it is. It depends on your task. I might need more context on that, but my off-the-shelf answer is yes. It should work. The interactive segmentation should definitely work. The automatic segmentation is a bit task dependent, but maybe if you want to explain, you could drop in a message or unmute yourself, however you prefer. Okay. Then the first step is always to do something what is called compute embeddings. So I have previously mentioned a few minutes ago that these models are kind of heavy and they take a couple of minutes to run. And the heavy component comes from the vision transformer. Without going to details, this is the big model which extracts meaningful features out of the image, which kind of resemble what an object looks like, what an object does not look like. Another question. Thank you for doing this. Perfect. Thank you so much for your question, Vasant. Yeah, so extracting features, giving them to the next backbone so that they can process it a bit more efficiently. So we compute the embeddings over here. And what you have to look over here is there's this blue progress bar in the bottom right, which kind of tells you how things are going. Again, Napari is not responding, but trust me, it's doing things in the background. And then once things are proceeding slowly, since it's a CPU laptop, things take a bit longer. Over here, there are messages which will pop up. Unfortunately, it disappeared, but you will see info messages in the console over here as well, or the terminal, that the embeddings have been computed with the light microscopy model of a particular size. Now we go back here and then try out a few things. So let's begin with point prompts. So here are different layers as we call, and I won't dive in too much detail into what layers are and these things. If you're curious about some things or they like, let's say bounce off, drop me a question. I can probably elaborate, but I would just assume a few things. If you want to know more details, we have YouTube tutorials for all the things I'm going through right now in a much more detailed way. Okay, so point prompts, we select on this layer. We select on this plus sign, which is basically adding points. We select here. Let's say zoom in an image, put a click over here, and then press on segment object. You see this square bracket keyword? This means this is something called a key binding, which you can also press from the keyboard. So for simplicity, I will just click on the screen over here, and this segments the object over here. This object looks exactly how I would like to segment it. And then there's an option called commit over here. So what this button basically does is currently it's a temporarily segmented object. Once you are confirmed that this is an object you want to segment and you want to store it in memory or in a new, let's say, structure, this is what is called committed objects over here, wherein the idea for committed objects is once you're confident that this segmentation is what you like, just commit this, the prompt goes away, and this segmentation is now stored for sure for you. Now this is done. Let's try another point prompt stuff. So let's see this big object over here. Or let's take this one because this is an isolation and I understand this way well. We click here, do segmentation, and do the commit workflow. This is again stored. Now let's go to box prompts, because as I promised, there is a point prompt wherein you put points and interact with the model. There's another mode of putting box around the entire object. So let's go to prompts and we add rectangles over here, which is the square option. Now let's zoom out and find a nice looking cell. Okay, this is a cell I understand as well. So let's put a massive box around this. Now we segment this particular object, and then we see that this cell has been segmented pretty okay, I would say. So we just press on commit and then store this as well. Now let's segment another one of these. Okay, another thing. So I made a mistake over here and put a bounding box. So let's say we put some random things here and there. And I'm like, oh no, this is something I do not want to do. So there's this feature called clear annotations, wherein the idea is if you made a mistake and you want to go back one step, not the entire steps you've taken, what you do is select on clear annotations and these boxes disappear. Remember that the segmentations you committed already will still stay there, but then things what you, let's say, did not commit yet will go away. And now let's put another box around. So let's say randomly, this is the box just fitting around a cell vaguely. So this is what it's segmented, but then I'm like, okay, this box is not precise. Could have been a bit better, but maybe let me try to improve it. So I put another point on top of the box and now segment the object. Now I see it's trying to improve this and it kind of did improve this a bit, but not by a lot. Let's say I'm happy about this object for now because I wanted to show you that prompts can be combined together. That was the message. And now I just simply commit this because I like the cell looking like this, for example. Now there's another feature, which is why I was leaving this big cell over here, which is called, let's say, freehand polygon lasso tool, wherein the idea is simple. You select on this tool, and then you keep on going and drawing around the particular cell if it has a very abstract shape in particular. And then you put the polygon lasso tool around this and then segment the particular object. Now, if you notice over here, the object has been segmented very well. It might look like it's fitting around the object, which is also in some potential kind of true, because the idea over here is we kind of trace a mask around this and try to segment using these masks. If you remember in the beginning, I explained it to you guys that there will be a point interaction, like a point prompt based interaction, a box, or a mask could be provided as well for more fine grained structures. And this is one of those examples. Now we commit this, and then this object is stored as well. We could do another thing, which is called batch prompts. Let's go to one area where I recognize cells right here. Voila. Okay, so what we do over here is select the point prompt layer, go to points, and then select this option, which is called batched. So it's just a checkbox. What this basically says in the tooltip is choose if you want to segment multiple objects with point prompts. So we put a click here, we segment the object. In this case, if you see me not clicking here, but the segmentation popping, that means that I'm pressing the S key binding in my keyboard. Over here, another object, another object, another object, another object, a few more objects in parallel. Boop. And then these objects are segmented in like a continuous round, for example. Now, let's say these are the cells I want to segment. I also like this cell. I will also segment this. And then once this is done, we go ahead with the same functionality as we discussed, which is the commit option. So we just press on commit. All of them are stored. Now let's quickly look at automatic segmentation. For this, I will switch off the committed objects layer, which you will understand in a second. So there is this functionality called automatic segmentation. Now, on a higher level, my go-to recommendation is you do not need to explore these hyperparameters over here. If you, let's say, want to get a good performance because this model is kind of optimized, or the not the model, but the hyperparameters are kind of optimized to do a good job on new images. And we just click on this without worrying too much about the parameters. So we click on automatic segmentation, and then there's a progress bar over here, which tells us a few details. Depending on your system, if you have a GPU machine or a MacBook, this is kind of fast. If it's a CPU machine, as you saw, it takes a couple of seconds. Now I close this toolbox over here, and then we see all of these objects have been segmented. Now we instantly see that the large cell I promised was a whole cell is kind of split into three. So it's kind of the automatic segmentation's fault that it didn't do a good job here, but we can rectify this. So we stick to automatic segmentation over here. Now we go to the pick mode, which chooses a particular color. So out of all three splits, we can choose one of them. And then there's a fill mode, which kinds of fills up or covers up them in all consistent colors, which makes this pixel precise as area as one whole cell. This is the idea over here. So this is how I made a patch fix. And this would be everything for the 2D part. Any questions so far? If that is not the case, then we go to the next part. Okay, so this is done. You can also save things. I'll quickly show you something. So let's say I only want to segment, like store the auto segmentation. I do not care about the interactive part. So I go to the auto segmentation, go to file, like after selecting the layer, and then save selected layer. This will open a console for me, which will allow me to save things in multiple file formats. I would honestly recommend going for TIFFs, but you can go for any and every file formats in general. Okay, let's close this because our task is done over here. Okay, this is task number 1. Now, what's task number 2? Let me quickly search up, or why to search up? I might have it here. Yes. Okay. So we have a task at hand for looking at light microscopy segmentation in 3D or electron microscopy segmentation in 3D. Are there any votes for either of them? Because I think if I account for discussions, we might have time for only one. And then I would want to give you guys room for chatting and asking questions. But is there any preference? Should we go for EM or LM? I can also make a toss or make a random choice, but if someone has a preference, would be nice to hear.
Let's make LM.
Let's make LM. Perfect. Wow. Okay. Okay. We'll do LM. I thought. Okay. Okay. Cool. Wow. Okay. Let's go for LM over here. And then I just simply copied this because I've done all the downloads over here to make this process a bit more faster. And I just copy this and then just hit enter. Since everything is downloaded in the format of this I2K folder workshops, then we just go ahead with segmentation. Let's see how this works. So what this part over here is doing is kind of loading all of these things and preparing us to get started with annotation right away. Because if you remember, there was a step which took a couple of seconds, which is compute embeddings. But in this case, we pre-computed this so that we can do this process quite fast. So assuming that we all paid attention to point prompts and box prompts, let's go ahead and try some things out. So the first thing would be trying to segment things in one plane. So we just select this point prompt, we select this, segment this, we segmented this. Now the problem is this is segmented only in this plane. This is not an object which is segmented throughout. And that could be a mess if I have to click every one of these. I can also show you, click this, click this, click this, But as you see, this is too messy. This could take a while, or you could easily interpolate throughout the slices if there's an assumption of the objects being isotropic or something, for example. But again, let's not go into details. I clear the annotations as I taught you because I want to show you something cool. So things can be segmented in a plane, but what if we could just put a click over here, segment the current slice, which is something what we did already. But can this one prompt segment all the objects or this particular object throughout all planes? So we go to segment all slice. Again, there are some heuristics over here. In a nutshell, they're kind of optimized on a model basis. You do not need to worry so much about them. We close this. We segment all slices, and there is a progress bar over here. It did something because the progress bar disappeared. Let's see. Okay, it was segmented for a few slices, but not so great. So what we do over here is put another point, segment the current slice, and segment the trailing slices. We see the progress bar has appeared again. Let's open this to see more details. Okay, just a second so that everyone sees this. Yeah. Now what we see over here is, okay, it could segment this very well, but the next slice is not. So I put another point in the few slices later on, and then try to segment this throughout. So the progress bar is trying to segment the object throughout all the planes. Let's see how that does. Okay, not, yeah, slightly bad for sure. But then we put a rectification point and segment all the slices again. And this is how you can improve the segmentation using, let's say, hopefully a few clicks. In this case, the object, let's say, contrast is not so great to do the segmentation with a few clicks. But then this is where the fun begins. If the model does not do a good job on your data, you just have to annotate a few planes since the training is in 2D only, and then annotate a few planes and retrain your own model. This natively would be a great segmentation experience for your task. Now, let's say how many planes did, oh, this side was pretty okay. Oh, interesting. Okay. Yeah, there were some mistakes over here. Let's put another rectification and say I will be happy thereon. Let's see. Ta, 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 ta. Okay, something happened slightly better, but let's say this cropped cell is the cell I want. Now, in this case, the functionality kind of works the same as before. You have this option. Let me close this console and let me go to the selector. There's this option of commit wherein this segmentation is currently still temporary. You have to store this or save this in a differently. So what we do over here is just click commit and voila, this has been segmented. Let's see how this looks in 3D. Might not be a lot of perspectives, but yeah. Oh, I see something. Okay, so the cell over here has been segmented in 3D. There are some, let's say, missing nooks and corners, which we could have improved, but maybe let's focus on the features right now and then understand the software. Okay, this is done. And then we can also try to segment other things with the box. So let's try that out actually. I'm also curious. So let's say I put a box around here. I segment it over here and I segment it over the slices. So how does that work? Okay, it is taking a couple of minutes, but this is yeah, good feedback at least. Okay, it did something. And oh, okay. It kind of in one direction did a good job. Let's see the other direction. Seems like, yeah, there are some mistakes. Let's fix them. Pa, 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 pa. Let's ignore this because I did something bad here. Yeah, in any case, if you guys have questions, whenever you see the progress bar, raise your hand or unmute yourself and ask. Because this progress bar usually takes a bit of time and yeah. Okay, this is done. Did it improve? Only the slice, not in the future ones. But let's say I'm happy about the segmentation right now. I close this console, commit this over here, and this is stored right now. Let's ignore whatever the artifacts are. Okay. Now let's try one final thing over here for us, which is called automatic segmentation. So we go to automatic segmentation, and then currently only, let's switch these off so that we try to see this part over here. And then let's run automatic segmentation for the current slice, because the idea is it will only do segmentation currently in the current plane. If you have to do it for the entire volume, we also see this in a second. We switch on this button called apply volume. So for this one, let's say if we switch this on and off, it did a good job. Some places it did a bad job for this long cell over here. But let's probably ignore it and see how it works for the entire volume. So we select the entire volume, we zoom out. The room is open for questions because this takes a few minutes. Automatic segmentation, you will see a feedback over here. I hope that is not too slow, but then are there any questions?
Do you have any tips for the fine-tuning? Or, I mean, it's a bit of a broad question. Or
Yeah.
also, is there, have you tried like just doing like lower rank adaptation versus like a bigger fine-tune? Any insights on that?
Yeah. So we have tried like the paradigm of parameter efficient and resource efficient fine tuning in its entirety, actually. In a nutshell, I go back to the first question. Like, since we really care in some, like, we have a follow-up work, I can also send this in the chat, actually. Good promotion. And it is basically on resource efficient fine tuning, wherein, let's say, we develop this software, it's going to take 30 minutes. Wow, we are not doing this. So let's say the idea would be you have a model for you, which does a very good job for, let's say, interactive segmentation. You got interactive segmentations for it, but for automatic segmentation, it's a bit poor. And let's say in like daily lives, we do not have GPUs at hand. And we want to like, let's say, train these models fast on low resources by, let's say, still watching a YouTube video, for example. And then over here, the idea is opening up resource efficient or parameter efficient fine tuning, wherein you retrain the model on your data to make it more knowledgeable about your data and do a better job. But if you see, the keyword is better, it's not doing as good of a job if you literally, let's say, retrain the model of all and update all the parameters. But if you become selective, it's a bit tricky. There are some gains which you have, but on a higher level, there's no clear outcome. It is a very data dependent question.
Yeah.
I dropped like a mention in the chart of our follow-up work, and this kind of explores this very in like extreme detail on like what is actually the problem over here of when we start talking about parameter efficient fine tuning, for
Cool.
example.
Thank you.
Mm-hmm. There's another question. Has your model been trained on other type of bright field images than mammalian cells? Bright field of plant cells? Yes. So there is this data cohort called plant cell, and we trained on, I think, the entire plant cell for root cells, for ovules, for cilia, and a few other structures. So, and these were imaged, some were with confocal, some with light sheet, but there was a wide range of it. I'm guessing that I hope that answers your question. But there was, let's say, a huge range, but not a massive range of data, because from our knowledge, PlantSeg is the only biggest plant cell cohort we are familiar with. So if you're familiar with any of these plant cells, I would be very happy to have annotated data. But the plant cells images look like fluorescent images, not bright field. Yes, that one you're right about, actually. They look more on the fluorescent side because one of them is light sheet. Another is confocal. I forgot the staining or any details about the root cells. So yeah. Bright field, can you give me an example? I'm very curious about what you're talking about. Or maybe I can search it up with you guys together. This will take a while, probably. I will close this, but we can chat about questions. Close. We can quickly quit, and go to this. Let me move this aside. Force quit. Yeah. Okay. Plant cells. Bright field. I'm very curious. So would you say this is one of the good examples over here? Because I think I am looking for one. Okay, perfect. I'm waiting. In case anyone has any other questions, please feel free to shoot a question. Happy to answer. I think I have a feeling of what you're talking about, if I'm not wrong. Sorry. Is the model trained on low magnification images on neuronal mitochondria? If not, how can I train the model using my own images? I don't think we trained it on neuronal mitochondria data. I do not remember off the top of my head if we had any. I'm guessing when you say this, you're talking about EM or you're talking about Super S? When you mean neuronal mitochondria. You would take an assumption until you answer and say EM. In that case, for EM data, you can really train your own model. Oh, even better, confocal. Cool. So for confocal, the problem always is the resolution in this case. Now that I think about this, but if you have, let's say, so for confocal, I would opt for semantic segmentation, which means that all the mitochondria will be segmented in one cell, for example. I wouldn't opt for instant segmentation because I have a feeling annotating mitochondria in confocal would be a bit too painful, I would say. So I would probably opt for, and I don't think it's even realistic to do that. But then if you're opting for, let's say, identifying the mitochondria or the mitochondrial structure in confocal images, I'd opt for the semantic segmentation track, wherein you just treat this as a foreground background task. And for this one, we have tried a few experiments, and Microsam does a pretty good job for this as well. Okay, there is an image here. Panel C, panel A. Security check. What? Okay, yeah, sure. Let me Google this. I might be faster. No, but there is an image. Yes. Is this the image? Can you confirm the ones on the right side or the left side? Yes, perfect. Easier than robot checks. Okay. I would say the right side ones are like have kind of an overlap with face contrast images, let's say on a morphological or a feature space point of view. What I mean by all of this is the model will recognize this quite well. On the right side, the model has seen a lot of images as B and D. So I would say all in whole, the major problem I would see is on the right side, identifying individual cells because the membranes are kind of overlapping with these fibers somehow. And on at least for the image B, it would, at least for me with the naked eye, it's very difficult. I'm not sure how Microsam will do. For A and C, I think it will do a good job because they kind of have an overlap of the plant seg roots data I kept mentioning. And now I see it's Arabidopsis roots as well. And you're trying to identify microtubules, for example, in this case. So I'm not sure how good it will do for microtubules, essentially, but these individual root cells can be identified very well, I would say. But give it a shot. If you're interested in microtubules, you might have to retrain a model, but then that would be kind of the direction to go. Just see if cell identification is your task. If you want to go further, let me read your question. But the plant cell images, yeah. So cell identification is your task. It should be like interesting to check out. I would say it should work, do an okay job. But it'll be interesting to see how it does on these bright field images. Okay. More fruit has research kit does not like me. I need to copy this. Okay, ResearchGate. What do you mean? I'm guessing you mean this one. Is this the one? Yes. Can you write another yes or no for A or B? Is it the plant leaf images or the A section? A, perfect. Um, so, a bit of a background. I'm not a biologist, so I will take some assumptions here. I'm guessing you want to segment these vesicular extensions on the outside. Is that what you want to do? These pipes? Exactly. No, Microsam would be able to identify this with some retraining, but off the top of my head, it will kind of struggle a bit because there are two massive structures over here. One is this main root hair. That's where the hint is. That's where the root hair is, and there are these fibers as an extension on the outside. In a nutshell, there are two targets over here for Microsam, or like pick up any model, cell pose, or anything in general. And since these are instant segmentation tasks, the first problem would be the discrimination. Model would not be able to discriminate between root hair and these fibers. So it's quite likely that the model will segment a lot of things, and then you might have to cherry pick, re-annotate, improve annotations, and then retrain a model. That would be my way to go to approach these kind of problems, wherein Microsam or Cellpose pick up any model, they do not work. Then you need annotations. If Cellpose annotations are good for you, or Cellpose or Microsam do a good job, take that as a starting point. Use Microsam to improve your annotations, retrain any and every model. So this would be the direction I would aim with. How many annotated images do I need to retrain? For this one, another shameless plug. Check the paper which I sent a few chats ago. That kind of explores the same question, but a hand wavy answer always for me is if you have 10, an important keyword, densely annotated images, this should do a good job. So as long as you have 10 densely annotated images, it kind of goes in the direction of literally getting a good solution for your task. Because these models kind of are aware of what they need to look for, but sometimes they're confused what they should be precisely looking for. And then that's where it comes from. Okay, Ricardo has another image. I hope that answers your question. Sorry, we quickly switched to the next question. Perfect. Nice. I mean, I think I am a robot. I'm not sure what's going on, but not sure. But this is an image which opened. That's good. Okay. These images. Wow. Too many things. Um, so I would say I'm guessing the green signal is like a membrane marker, and the red is an uptake of some sort of some compound. I'm not sure, but I'm guessing the idea here, what you are interested in, or the question you're asking is how to segment individual cells, right? And then you want to do something. Bingo, awesome. Then the idea would be, if you remember, I teased an idea of the object classifier. This would be literally perfect for this. Because the idea for that would be you use microsam to do instant segmentation on these membrane-bound structures over here, which are cells in our case, and then use object classifier to classify the cells on top, saying that these, I will say random things now, these cells have an uptake, these cells do not have an uptake, for example, of this particular compound. And then you have two outcomes at once. You have instant segmentation and object classification on top of it. And then you can do single cell analysis from there on for like a lot of things, for example, if you're interested. So that would be my 2 cents for your question, if that answers your question. Perfect. Awesome. I will still be here for a while in case you guys have questions. I would be very happy to answer. Do you think it need much computer power for that? I am guessing you mean just the annotation part, not the retraining part. Do you mean just the segmentation and annotation? Aha. For this one, you do not need. I'm again assuming these are 2D images. And I mean, if they are multiplex, like high res images, They should take a couple of minutes to run the first step of compute embeddings. But then once you've done this only once, you can literally rerun it and keep on annotating. So it shouldn't take more than a couple of minutes, I would say, optimistically speaking. 2D, but like 3 GB. Jesus, what's the big depth? Are we playing gigapixel resolution here? What are we playing with? Okay, what's the big depth? That would also be a big influence. Do you know if it's 16-bit, 32-bit, 64-bit? What are you storing it as? Yeah, that's what explains the 3 GB. Okay. Um, In this case, what I would do is do this in 2 stages because you have also like 4 channels. Totally fine. As long as you are interested in cells, just focus on three like important cells. Sorry, three important channels which kind of express the membrane or the cell. And this should be enough for you because this is how we make a choice in multiplex images, for example. Now coming back to the main question, actually, you have to do something what is called tiling-based prediction. If you remember the tile size and halo, those are very important because otherwise, what MicroSam or literally all models will do is take the image, squishes down, and see random things, which is not what we want. We want to see the same field of view as the one in your on our screen over here, which is look at the cells very carefully, a bit more closer, and this is what the styling based prediction does. Zooms in on one frame, takes a tile of say 512 times 512, segments this, goes to the next tile, goes to the next tile, stitches them back together. That's the idea. For this one, you have to do this in 2 stages, which is something I mentioned. The first stage would be compute the embeddings on a GPU system. So find a laptop which is either a MacBook or has a GPU. Like if it's a MacBook, should have these M chips. They are very good and they kind of, I wouldn't call it replicate, but kind of go in the direction of accelerating these deep learning architectures. And the idea would be you can run these very fast with these M chips, for example, or get a GPU at hand so that you can do the first step computing the embeddings. You have a RTX 2060. I think that's good. For this data, this is pretty good, I would say. Yeah. So this should do a good job for you once you have done, and you can literally do this in one laptop then if you have RTX 2060. So that should be the way to go. Would you talk briefly about how would you train SAM for semantic segmentation? I can look up an issue because we had some people wherein we wrote a script for them. Semantic segmentation, semantic segmentation. Yes, incorporating. This is for me. This is for me. Challenge semantic segmentation trainer. Oh, we have a semantic segmentation trainer. Wow, I forgot that. Okay. Give me a second. Semantic, there were some problems. I gave them a script. You have an example script now, actually. So over here, this is a follow-up work wherein we kind of wrote a script on top to do semantic segmentation with essentially SAM. So SAM or microsam, or in this case, medicosam, which is for medical imaging, is the idea. And you can do foreground, background as a task, for example, wherein you do not care about identifying each objects individually, but you can do this. If you go to medicosam, I'll leave a link over here. If you go to examples, you will find it right over there, actually. Semantic segmentation right here. And this should be like a direct adaptation because our models are cross-compatible. So let's say if you're doing this for microscopy, you just have to use VITB LM, which is like microscopy, and this works out of the box. You do not have to install 10 different things or do 20 different things. You just, everything is just cross-compatible quite conveniently. Yeah, perfect. Okay. Pa, 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 pa. Where were we? We were. I did I miss that? No. Yeah. Let me open Microsam so that I can walk you through a few things if there are any questions. Okay. Any more questions? This is your opportunity. I mean, you have more, but I mean, in any case, you can reach out to us in our GitHub repo. I try to stay as much as active as possible. But if I don't give you an answer, you will get an answer from one of our team members. If not, definitely Constantine will respond to you. So that is not an issue. And like hit us up with an email if there are some, let's say, private images you have which you do not want to share here, or hit us up on ImageSC. There are several methods to communicate with us.
Maybe two more questions.
Go for it.
The first one, what's the main differences between, let's say, like the earlier releases of Microsem and your newest release? Did you like retrain the model or yeah?
So you mean a release which is in production already, or the
Mhm.
Microsam 2 thing which I teased? Which one?
So let's first say the newest one that's out in production and then what you're doing right now.
Okay, so the newest one, there's nothing really different compared to, let's say, the videos you will see or what we have also talked about before with you, Philip.
Yeah.
There's nothing new. It's just we have a bit more data over there. If you ever are in doubt that, okay, they are doing something funny, I do not understand, go to our
Yeah.
docs, go to data sets, and everything is documented on which model is trained with what data, how much data, what is done.
Cool.
So let's take L4, for example. This is kind of trained on 14 different data sets for light microscopy V4 model. All the data sets are mentioned over here. And we also trained the EM organelle model on a few data sets over here because this data set, let's say the EM direction of annotating organelles is not so populated compared to LM. But then these data sets are, let's say, versatile slash big slash both enough for us to do an okay job for new data, for example. So this is the current release wherein we found a tiny bug in one of the model strategies or modeling strategies and convolutions. I won't dive into the details, but the idea was simply there were some artifacts being picked up. We had another paper, follow-up paper. We identified the problem, fixed it in Microsam. That was the
Okay.
idea. I can send you the paper as well.
Yeah,
Wow, a
cool.
lot of promotion for me. I love that. So this was presented recently in ICCV. This was a paper with Anna Kreshuk's lab. Let me send this over
Mhm.
here. So the idea was, in a nutshell, there are artifacts, let's say, which are popped up, and these artifacts could pop up for several things. In this paper, we explored normalization, but in our cases, we were seeing it for upsampling strategies. And
Mhm.
we fixed it over there. But then, like a shameless plug for this paper, not so related, but might be interesting on what kind of artifacts could come up with these foundation models or task specific models, for example. That is one thing. The next question, Microsam 2. So with Microsam 2, I will quickly take you here. The idea is currently there are models. So let's say UNet, for example.
Yeah.
I don't want to take specific examples. So let's take a UNet. You have to train a UNet for a 2D task or a 3D task. You cannot use it across both tasks. So if you trained a 2D unit for 2D models, you trained a 2D model for 2D data, you have to use it on 2D or use it across planes and stitch it back.
Mhm.
Now, if you use a 3D model, then what you have to do is somehow have to have a 3D data. You cannot use 2D data in a 3D model. It's just, let's say, as a construct of an architecture, not possible.
Yeah.
And then always there are problems. You don't have spatial context in 2D models, and you cannot reuse this for, I don't know, confocal images, 2D confocal images, for example, a 3D model. And finding a mid ground is always difficult. So what a lot of methods like CellPose, ASK, Stardist do is that they do a post hoc post processing on top of it so that you
Yeah.
can get better reconstruction of an outcome, but inherently are still a 2D model. Now, what we want to do with Microsam 2 is get away from this and do universal segmentation, wherein the same model pre-trained by us will allow accepting 2D images or 3D images. For 2D images, it would process as exactly as a 2D image. For 3D images, it will take the spatial context in the learning heuristics and give you a segmentation output. This is
Mhm.
kind of the high level goal we are trying to achieve over here. And
Cool.
we want to do this not just for automatic segmentation, but for interactive segmentation as well in a learnable strategy. So until Microsam 1, these are with a post hoc post processing. Microsam 2 will or is really focusing on, and the manuscript is in work in progress right now. The focus is really to make this a learnable strategy so that we can have the context in mind. Because, well, if there's spatial context, why throw it away? Just
Yeah,
consider
exactly.
it
Yeah.
and learn on it. That's the
Mhm.
idea. And we are
Yeah,
working on
cool.
this right now. Yeah.
Cool. Thank you.
Perfect. Sure.
And a last question, if I may, what would you suggest to use as validation metrics, or what have you used? Just IOU or?
So there is a metric which is called, I would quickly introduce you to a package which is called
Mhm.
ELF. And I will leave a shout out for ELF. It's developed by Constantin, and we maintain
Okay.
this
Yeah.
actively in our lab right now. The idea for ELF is simple. We kind of want to standardize all of these things of having a metric, having like a standard way to communicate numbers. And what we kind of presented with Microsam 1 is something what is called segmentation accuracy, which was taken from the Pascal Walk Challenge. And over here, if you look at this into detail, you will find mean segmentation accuracy and how it is kind of inspired over here, which is, let's say, a very difficult or a hard criterion because it's mean of accuracies over IUU thresholding from 50% to 95%. So taking a mean of this as an outcome is what we represent as mean segmentation accuracy.
Mhm.
In a nutshell, it's a very hard metric because you are considering IUU 95 as a criterion for your mean, which is, let's say, a bit rude to your segmentation method, but it
Yep.
is also fair to consider hard criterions as high as 95% of an overlap, for example.
Mhm.
And then this is what we do. We present all numbers in Microsam 1 on this. And we are very confident, like in our lab, all papers like which we are a part of, we kind of follow this track for instance segmentation. For semantic
Mhm.
segmentation, it's dice score, the obvious thing, having an overlap or checking up classic overlap. And in Microsam 2, we would kind of focus on this as well for instance segmentation. So if
Cool.
you want to go in the direction of having a standard, let's say metric, I would highly recommend looking into the Pascal Walk Challenge. It kind of standardizes lots of things.
Yeah.
And the DSB challenge kind of re-implemented this, and we have kind of re-implemented taking the best pieces of the DSB challenge, the data science bowl for nucleus segmentation, and the Pascal Walk challenge to get a, let's say, unified metric for us, which does a good job so far.
Okay, nice. Yeah.
Okay, 5 more minutes, everyone. Any more questions? We can also take more. That was just a warning sign. But any more questions, everyone? Uh, Thank you so much for being here. I mean, appreciate it. Mhm. Okay, if that is not the case, then everyone wants to take an evening off. In that case, I would say we pause over here. In case you have any questions, just hit me up on ImageSC, GitHub, by email, everything works. I am the fastest on GitHub and ImageSC emails. Let's say it's the usual academic thing. But if you have any questions, just hit her up. We can chat, send us a few images, corresponding labels, and we can figure out how to solve your problem a bit better. Sounds good? Thank you so much, everyone, for joining. I hope to see you guys next year at in-person I2K. Perfect. Have a nice day ahead. Thank you so much, everyone.